on a highly successful trajectory. As well as being such an investigator, she also performed pioneering work that really put structures in the deep brain on the map also, as being complex and important for making us who we are. She studies the neural basis of learning and action strategies relevant to decision-making, making and breaking habits, affective state, and brain disorders. And her talk title, An Ancient Brain System for Modulating Mood. Dr. Graybiel.
Thank you very much. And many people have talked about Pat and Lori. And I remember the first time I saw you two and then met the family. Thank you for being here. And you all made such a deep impression on me. You set the whole spirit of this place. You really did. And while you, Bob, talked about looking over the shoulder, I didn't have that experience. My experience was that every time, Lori, you too, every time I saw Pat, he had a smile on his face. He was completely interested in everyone, and every time he saw someone, it was as though they were the center of his attention. He asked what they were doing. He made us, each one of us, feel important as a human being and as a scientist. And the spirit that the McGovern family, Lori and Pat, and the family made was formative in this place. And I think this spirit has been shared by students and postdocs, undergraduates, and actually the Institute and many others who see this place as special, as full of energy, as looking to the future, as creating, and as at the same time as having some feel of community, of collegiality, and of sharing. So thank you, Lori and Pat. What I want to talk with you about is how we make decisions and how those decisions influence our action. This is actually almost a miracle that we can intend to do something and then do it. And this looping circuit is highly influenced by motivation, and somehow from that comes music and movement and thought. And central to this whole complex, wonderfully dynamic action of our brain are the deep structures of our brain. You haven't heard much about them today, but underneath this beautiful thinking cap that we have in the neocortex are structures that are, when this loop is broken, unfortunately, responsible largely for the extrapyramidal disorders. We've talked a little bit today about Parkinson's disease, but also neuropsychiatric disorders. This wasn't known until a few years ago, and one by one, these disorders were found to have some component related to disorder in the basal ganglia, and especially in the region I'll talk about called the striatum. And this same set of circuits related to the basal ganglia are responsible for addiction, as I'll come back to the effects of stress. So what is this system? We're going to talk about the fact that we assign values. We assign values to what we see, to how we act. And eventually, the neocortex projecting down to these deep structures, I'm showing you the striatum, these structures, through their outputs, influence the selection of the movements or moods that we will have. And the system is beautifully modulated by the dopamine system in the midbrain. I'm sure you all know about dopamine from Parkinson's disease. You may know it from studies of schizophrenia. And the thought is that this region can send, whoops, I'm really messing up with this thing, aren't I? It's okay. This system can send reinforcement signals up to, what am I doing? Now I've turned it off.
Push the bottom button.
Bottom button. Got it, okay. And then the top button without hitting the advance button. This is an intellectual feat of no small proportion. So, so there is the system. And let me say that the thought in the field is that in the neocortex, we find circuits that have to do with evaluating what to do. Then down in the striatum, we think, is it worth doing? That circuit does. And then down here in the midbrain, the dopamine system, which asks the question of what reinforcement should be given to this system. So the whole system tells us much about, as I say, action and thoughts that are going to come. Now, deep in this system lies this funny-looking structure. It is, you're seeing a slide that shows a cross-section through the striatum. It's stained for acetylcholine, a very important neurotransmitter in our bodies and brains. And it's partly funny looking because it has these little structures like there and there and there and there. And these, we ended up calling striosomes for striatal bodies. And although they look a bit funny, what I'm going to say is that in an amazing way, they turn out to be absolutely central in this amazing pathway that influences our actions. Now, if you ask yourself, how can that be? The very first thing that we found was that if you look at the brain of a monkey, these regions you can look at with tracer methods, and it was the blue region here that especially projected the striosomes. And the reason I'm emphasizing this is the following. Here is a scan of the human brain in a person with depression, and here you'll see that same spot is just in front of the corpus callosum, just like this. Now, if you look at someone who craves cocaine because he's a cocaine user, is the same region. Now if you look, this is an amazing study done in Germany, at migrants who have come to Germany, to a city, they become incredibly anxious. They've never really been in a city. They're so anxious that they end up having to have therapy. So these are regions which putatively might project here, and I'll show you some evidence for that. Now, gee, I wish I could see this better, but let me show you this system in a mouse. And I'm going to show you the system with the front of the brain on top, the bottom of the brain on the bottom. So the brain, which normally is here, has been tilted like here, and you'll see why. Here it is, shown from the back to the front, and now we've genetically labeled those striasomal neurons, and what you can see is this labyrinth made up of the striasomes that show up as red, labyrinths that fill actually a fair amount of the striatum. So we now have a system that starts out with this evaluation cortex, somehow targets parts of it, the striasomes. And the special thing, the amazing thing about these little striasomes is that they are the primary regions in the striatum that have the privilege of projecting directly to dopamine-containing neurons, which are essential for our normal mood, motivation, and movement. So the question that we came up with is, how do we deal with this system? How do we understand if it's a decision-making system, what's the decision and what controls it? And so what we decided to do was, in animals, try to mimic one of the major categories of abnormality shown by patients with depression and anxiety. And here's what this system does. You see him here. He's trying to decide whether to approach or run away. You see his friends are already running away. And here he is. And that is called approach, avoidance, conflict, decision-making. And as I said, what am I doing? I'm so sorry. This doesn't usually happen to me. This system is tremendously present and highly vulnerable in states of anxiety and depression. So what we did was we decided to mimic that task, as I said, in macaque monkeys and later in mice. And so we could look at the amount of benefit, if we look at this in terms of a cost-benefit mechanism, here's the amount of benefit, y-axis, the amount of cost. And so these are the decisions shown in these two symbols made by the animals. Now, if there's a lot of benefit and very, very small cost, the decisions will be down here. If there's a lot of cost, very little benefit, the decisions will be over here. So what we could do was define a decision boundary between these two behaviors. And that was really important, because then what we could do is see whether by manipulating the brain we could change the behavior of the animal toward approaching more or avoiding more. And so we decided we would do that in that blue area. And what we found, and this is the work of Kenichi Yamamori in our lab, what we found is that if we stimulated that blue region, we got a very strong increase in avoidance. The animals acted as though what was coming in the future was less good than it would have been had the stimulation not been there, as though they were pessimistic. So this phenomenon was blocked by anxiolytics used to treat people. For example, the effects of diazepam. It was dose-dependently blocked, this entire effect. There wasn't any effect on our control approach test where everything was good. And then, most recently, we've applied with the Pizzagalli lab at McLean Hospital this same kind of task in humans, normals and people with depression. And the really nice thing is that in the study, we've used precisely the same stimuli as used in monkeys, and everything we could do, we did to make the situation similar to what we'd done in the experiments. And what you can see here in the title is that this region, which is right in front of the corpus callosum, you've seen it over and over again, this region was active when the people felt that the offer was aversive and when they actually avoided. So this gave us the first hint of a commonality across many disorders, and even could be seen in the tasks that we were using. So this is an example of stimulating the striatum itself with similar methods. And you can see this decision boundary. And when the stimulation, so here are the avoids, red, the approaches in blue, the decision boundary. And when we turn the stimulus on, you may notice there's more red. And then when the stimulation went off again, it went back to the original decision boundary. We were shocked when we looked at the monkey the next day. Here's just the next day. Here's the stimulation off, and yet there are a huge number of avoids. Here's the stimulation off, a huge number of avoids. And again, finally, it began to go on until in the third session, it normalized. What that means is that whatever we did, when we applied microstimulation to the striatum, and you saw it before in the blue zone, we were getting an effect that could last. That was very, very surprising and interesting. And again, this was blocked by diazepam, and then again, there was no effect in the benefit test. So we wondered what this could be and why it would be only present for these negative effects. So we asked, could this be a proxy for pessimistic decision-making, for something related to depression and anxiety? And for that, we really felt that to get a better grasp of this, we should change to working on the mouse, where the methods, as you heard all day, are more advanced and actually applicable in a better and more straightforward way than in the monkeys. And so we did that. And here you can see actually a blow-up of a single striosome. And what we've been doing now is to try to get at the systems level activity of the system and the molecular specificity of it. So we turned to an old classic psychology type T maze used for, I don't know, 150, 70 years. And so what we did was to set up a very simple cost-benefit task. These mice love chocolate. So here's a little pot of pure chocolate milk, and the problem is we paired it with very bright light, and they hate bright light. On the other hand, if they would go this way, the light would be dim. On the other hand, the cost would be the chocolate was really diluted. In fact, we did parametric tests on how much to dilute the chocolate. And so a normal mouse will, of course, avoid the light a lot, most of the time. And then what we did was we looked at this behavior, and then we looked at a microcircuit which connected the neocortex to striosomes, and there was this interneuronal system that could change, we found, the whole dynamics of this circuit. And let me show you how we found it. Here's the normal situation. The normal mouse will mainly avoid the light and give up the high benefit, go that way. And here's what happened when we manipulated that circuit, and especially those interneurons. What happened was, to our amazement, now the mice mainly ran the other way. They're running into something they do not prefer, something they normally would run away from. So what we found, in essence, was that if these little striosomes, which I showed you as red cells in the labyrinth running through the striatum, if they were turned on, they were active, then the mice would approach aversive flight. But if they were turned off by our manipulation, which is often genetic, then the mice avoided the bright light and acted normally. So we asked ourselves, now look, if this is somehow related to something like anxiety or depression or pessimism, that's like a state change. It's like a whole different thing. You get up in the morning and you just don't feel good. Things don't seem so good. I think everybody has experienced that. Most of the time we want to wake up happy, full of optimism, like Pat and Lori, but there are those days. And so could these striosomes, or that circuit somehow, be kind of like a switch? And could that switch be mediated by those striosomes, which would be amazing? And then we asked, now look, if it is a state change, then it should be able to change it just by experience, without manipulating the brain. So here's what we did. We started with mice who had not had stress, and then we applied to those same mice very mild stress. For 2 weeks, once a day, they sat in a little tight cloth. And I think of it as going across the country, you know, California to Boston, Boston to California, but you got a middle seat, right? And by the time you're done with that flight, you know, you're cooked. So then we gave that gentle stress. And let me go back. Here's a normal situation before the stress. Now look how they changed the direction. No thing was done to the brains of these animals. Nothing. The only thing we did was to give 2 weeks of this very mild chronic stress. This experience alone can switch the circuit dynamics. You heard from Ed Boyden how important dynamics is to these circuits. And what we found, I won't go into, is that millisecond changes in the dynamics are important for this switch based on experience. So then we asked, well, if we have the switch and it's experience dependent, is there any way to rescue it, to get rid of it, to block the effects of stress? And so, let me go back. What we did was to manipulate that circuit, and I hope that you can see that that manipulation of the circuit completely blocked the effects of stress. So we were very excited about that. We still are excited about that because there are so many conditions, partly in real life and especially in disorders, PTSD, in which we need to help have people reverse the stress under which they are. And we may have found one circuit, there may be many, which can handle this, which is very useful if we're thinking of eventual therapy. So now what we turned to was this last part of the circuit. I've talked about this, these connections. We've talked about what's going on in here, but I told you that striosomes are special in the fact that they are just about the only neurons. They may not be the only neurons, but they are the majority of neurons apparently in the striatum that can project and therefore influence those very, very important dopamine-containing neurons, which absolutely can change our mood and our impulse to move. So I'm showing you here a little bit about what the dopamine-containing nucleus looks like. And now look at this. Isn't it beautiful? These are the dopamine-containing neurons, all stretched out in the substantia nigra pars compacta. And the white zone and pink, the green and the white, those show the striosomal fibers that have come all the way down from the striatum to the dopamine-containing nucleus. And they probably synapse, almost certainly synapse, on the dopamine-containing neurons. Now I'd like to introduce what we've called the striosome dopamine bouquet. And when we first saw this, we saw it first without, but then with Ed Boyden's expansion microscopy method. And you'll see these are like roses, and they're stems. And when you see the green and yellow, these are the striosome fibers climbing up the stems, making contacts down here, and finally reaching the level of the roses. So now what we're doing is asking whether these striosomes could turn on and off sets of dopamine-containing neurons. So they could perhaps change these moods. And so we're trying very hard, and it's difficult, to modulate parts of that circuit. We're doing a great deal of mouse engineering. I never thought our lab would be into that, but we are, seriously. And what you can see over here, but it's not blinking and it should, blink. Blink. Anyway, here, there, you can see some blinking. Now we're doing 2-photon microscopy. First at the level of striosomes, we can see them now. Because of the method we've used to manipulate them, we can actually see these little patches. So we can see the activity by calcium imaging. So I've told you now about 3 legs of this circuit, and do you see it runs all the way from bits of cortex that we know are involved in controlling mood, motivation. They're abnormal in patients with many different disorders related to these mood and motivation systems, running down through here all the way to the reinforcement system. And let me tell you one more thing. If we look at this system and we look at what chemicals could influence the system, the first thing to note is here, opioid receptors. You know, the opioid problem in our country and others. These express the receptors that these drugs, like fentanyl, are hitting. This system also is very, and now I'm talking about striosomes, very rich in particular kinds of serotonin receptors. That's a new finding. Well, serotonin receptors of the kind I'm talking about are targets of SSRIs, which are given to people who have problems, among others, with anxiety. Well, what about cannabis? If we're getting going on opioids, let's go all the way and look at marijuana and stuff like that. And we've just found that these receptors, guess what, are heavily encrusting the bouquets. It's quite amazing. So in this system, I mean, we're only at the start, but look at this list. So this again makes us feel this is only one set of circuits, but looks like it might be really important. So I want to close by coming back to the spirit that's been created by the McGovern family and by Pat, coming every time and looking in the face of people. I've seen it happen, or not seen it, but I've felt it as he looked at me, and the inspiration that he gave and that you gave, Lori. And now I know that you have given me. It's invaluable in my life, and it's invaluable in science here. So let me thank many people who have, through the time that you have started this institute, and I'll tell you, every single one of these people, everyone is grateful to you and Pat. Thank you.
Thank you very much, Dr. Graybiel. Do we have a question from the audience?
So that's very interesting. So where does cortisol, the stress,
Pardon me?
where does the stress hormone cortisol fit into all
Isn't that interesting? So we're not that far. I mean, we have this sort of thing in mind, but we had to, what we decided was, let's see if we have a circuit, now we can manipulate it. And so we have sort of a list. And the very first thing we actually are working on is using CRISPR to affect, knock out, knock in these receptors. Emily, I don't know whether Emily Hicksta is in the room, but she's in the beginning stage of CRISPRing that very particular serotonin receptor, and the reason is, boy, do we know it's important, and it's such a particular receptor, but we'll get there.
Okay, because
Yeah.
I understand it stays in the system for 48 hours, and you showed that graph where
Yes.
you press you put the stressor, and you went back, and you found that even afterwards, that dynamic was still there. So I was thinking maybe that's related to cortisol.
It could very well be, or ghrelin. It's an excellent question. I wish we could already answer it, but we'll try to. Any more?
Thank you so much.
And thank you.
Okay, good.